So it's great to be here. Actually, it's a little bit of a white lie. My wife and I just got back from the Caribbean, and so you can see I managed to put long pants on, but haven't been able to put the long sleeve shirt on for this weather. But no, it's good to be here, really. It's a great event, and it's great to be here. So as Jeffrey said, we're going to talk about, I think you said early, not so sexy anymore. I think, wasn't that what you were talking about, library prep?
Yeah, maybe a little more commoditized.
Yeah, a little more commoditized. So we'll try to take it back and make it a little bit more sexy again, and hopefully not any sexy dancing or anything like that. So why care about making libraries from super small fragments? I mean, the bottom line is everybody's talking about large PacBio and larger fragments, long-range sequencing, and all that stuff. Well, there are definitely some reasons in some cases where doing short fragment sequencing is important, or ultra-small fragments. And so those come through in samples that are degraded or naturally small. So formalin, fixed, paraffin embedded, FFP samples, they're horrible. If they're old, they're usually super degraded. Ancient DNA, we're talking about extracting DNA from really, really old bones. Liquid biopsy samples, so cell-free DNA. This is a sexy term. This is the big thing in molecular diagnostics, cancer oncology work. Metagenomic samples, small amplicon pools, forensic samples. All of these can have, at least in some cases, very, very small fragments that you might want to sequence. You want to extract that data if you can. And then finally, there's other applications that may benefit, such as ChIP-seq. Okay, so chromatin immunoprecipitation. Everybody wants to do ChIP-seq because they want to narrow down their binding sites of their proteins or their chromatin in vivo in their cells. And so there are methods out there, such as micrococcal nuclease digestion ChIP-seq, where you make very, very small fragments. And the ability to make those good, high-quality libraries out of those chromatin IP samples is really important. All right, so what I want to do now is just take a minute and set up the problem. Okay, so we talked about the goal here, which is to make ultra-small libraries. What's the problem with the current systems that are out there? Okay, so here's a typical Illumina library prep schematic. So you start with your genomic DNA, it's fragmented, the ends are repaired and made blunt, and then they're A-tailed, and then you adapt, or you ligate on a Y adapter, shown here, and then in best case scenario, you get all your fragments with adapters at both ends, which we all know is never the case. But this is ultimately the end material, and this is a PCR-free library prep. Most people, and for the applications that we just talked about and the samples that we just talked about, people are doing a PCR step. They're adding in PCR with primers that bind to the P5 and P7 sequences and end up after PCR making completely double-stranded library fragments. Pretty simple stuff, basic stuff. I think this is the boring part that Jeffrey alluded to. Now there's some issues here, and the first one is that you don't want your final libraries to be contaminated with adapters or adapter dimers. And so if you look at the adapters, this is a standard single index Y adapter. It's got a 58 base pair or base oligo and a 63 base oligo annealed, about 12 bases of complementarity there, about 12 base pairs. So they're about, 60 bases long overall. Adapter dimers are when these molecules actually get ligated together. Then they become about 120 bases in total with about 24 double-stranded base pairs. And they do run larger on a bioanalyzer. You're going to see some bioanalyzer data. They run larger than they should on a bioanalyzer because of their single-stranded nature. And then after amplification of these, if you have any adapter dimers, you can end up with these amplified adapter dimers, basically perfectly double-stranded now. They're about 121 base pairs, 120 base pairs long. And that's a bad thing. You don't want these molecules making it onto the sequencer because they can ruin your sequencing data or severely limit the data. So here's the problem. So if you're trying to make ultra-small library fragments, or fragment libraries, if your adapter dimers are 121 bases, your amplified adapter dimers are definitely 121 bases, base pairs of double-stranded, and then you make a 25 base pair all the way up to 180 base pair insert, you'll see that these products are 121. The first one's 146, and of course it's going to be a range depending on what type of library you're trying to make. So separating these molecules, for instance, from these adapter dimer that have been amplified is really difficult. Most of that's done with AMPure XB beads or similar beads that are on the market, and it's very difficult to be able to separate these. And I'll show you in the next slide an example of that. Right, all right, we talked about all that. So here's a bead size selection using AMPure XP beads. And of course, the key here, if everybody knows, is the ratio of the beads to the samples. That dictates what size fragments are going to be pulled out of that sample. So in this example, this is the sheared DNA that we use. We enzymatically sheared it to make it very, very small. These are all run on a bioanalyzer. And then we captured the samples with 1x, 1.52, or 3x AMPure XP beads, found standard protocols, washing, whole 9 yards. And then ran them on the gel. And so what you can see is as you go up in bead to sample ratio, you get smaller and smaller fragments that are captured, okay? And you tend to lose some of the larger fragments, okay? So the key here is being able to separate the good stuff in green from the bad stuff in red, okay? So here's an example of primer dimer with a standard Y adapter. And this is amplified or unamplified. It's about 121 base pairs. And then if you've got a 25 base pair insert, you're at 146, 177, 221. I didn't put them all in here, obviously. But what you can see is there's no bead concentration, bead to sample ratio, where you select just the library and not the primer dimer. So it's this is just illustrating how difficult it is to really separate these two when you're making these libraries, okay? So this is our solution. And this, we've done this for many reasons, but what we've done is we've created, instead of a large Y adapter, we've created a mini adapter. And this is part of our NxSeq UltraLow DNA library kit. I won't talk about the kit in much detail, except that it's very good at making very good libraries. We can go down to 50 picograms of input. It uses PCR to make amplified libraries, but it's really all developed around this mini universal adapter. So all this part looks the same, but then we've got this universal adapter. We call it it's also called the mini Y. It's about 26 bases long, and that's going to help us when we talk about small libraries. So what you end up with after the first start of the library prep is you end up with library fragments. They look much like what we just talked about with the standard Illumina prep, but they're not sequenceable, okay? They're not complete. They're not ready to go on the instrument. So that's where the PCR comes in. We use indexing primers and a primer set that, in this case, is just a single index. We've got dual. But you'll add a P5 primer or P7 primer with the index. You will amplify these first with the P7 primer. It'll bind, make an intermediate PCR product. That's then a template for the P5 primers to ultimately make the completed PCR product. So it takes 2 cycles to make a fully sequenceable fragment here. All right, so what about it? What's the big deal? Well, the big deal is that it really helps in separating the good stuff from the bad stuff, okay? So again, bad is in red and good is in green, and this is just an example with a 25 base pair library. And I'll show you data that this is actually doable, okay? So as I said, the universal adapter is about 26 bases long in total, okay? So an adapter dimer is 52 bases. When it's amplified by PCR, it looks like a full-length Y adapter dimer that's been amplified. So it's 121 bases. So if you had a 25 base pair insert here with the adapters, the universal adapters, before PCR, it's going to be 77 bases. After PCR, it'll be 146. These are still difficult to separate. So what's critical here is being able to separate these 2 molecules. So you want to get rid of the adapter dimers after ligation before you do the PCR step in that slide, the previous slide that I just showed you. And you can see that we're down here, we're right on the edge, right, of being able to do that with leaving the adapter dimers behind but capturing the universal adapter ligated fragments, okay? And then if we can do that, once we amplify, everything's good. It's a lot easier when you look at a 50 base pair insert, just as another example. Oops, sorry, where did it go? Oh. Anyway, there should be lines there. Basically, here's your pre-PCR library fragments here. So they're much better separated from the adapter dimers, okay? And then the amplified is still there, and the 50 base pair is larger. So these are much easier to actually clean up and separate the bad stuff from the good stuff. So here's what we recommend. In all the data that I'm going to show you, this is our library prep. You start with N repair and A tailing. It's a 40-minute reaction. Add in your universal adapter and ligase, go 30 minutes. Then you've got your first bead cleanup step, which we recommend. First bead cleanup, then 5 minutes to set up the PCR, and then you do a cleanup and a size selection. We've got 2 here at the end just because our ligation buffer is viscous and it can actually inhibit a good size selection, so we do 2 we do a cleanup and a size selection. Basically, the cleanup's a buffer exchange. And what we recommend when we're talking about these ultra small fragments is we found we've been able to do a 2x bead cleanup step before PCR, 2x after cleanup, and then a 1.5x size selection at the end. Okay? And that all the data that I'll show you uses that type of protocol. So here's an example. So the first set of data that I'm going to generate, the first stuff was all theoretical, just showing you where everybody lined up on the bead traces or on the cleanup steps. What we did was we made a series of synthetic oligos, double-stranded oligos, and we used those to make libraries. We didn't sequence them, but we used them, and we analyzed them in the Bioanalyzer to see how clean they were. And what you can see is this is the worst-case scenario. We've got a 20 base pair starting fragment. This is after ligation, but before cleanup. You can see you're starting to see some extra material. The dark red bands represent the good stuff. I've lost a lot of arrows. I don't know where they went. And then after ligation, you can see we end up with just the nice universal adaptor ligated materials in the Bioanalyzer. And then after PCR, we start to see some of the PCR junk that shows up. And then after PCR cleanup, you can see we've got a very nice library band and no contaminating adaptor dimers or small molecules. Okay. And this is the same thing with a 90 base pair insert. Really doesn't matter. It's easier than the 20 base pair insert. So let's look at all of them. So we did a whole series of fragments. So we went from 10 base pair DNA inserts all the way up to 90 base pair DNA inserts. This is the starting material analyzed on the Bioanalyzer, and these are the final libraries. And what you can see is it's actually there. There's a 10 base pair band there, but it's really faint. So we've probably lost a lot of material. It was just too far down on that size selection or bead cleanup steps to be actually capture most of the material. So we suggest that we can actually do 15 base pairs and larger with these libraries, and you can see that they're nice and clean. Okay, there's not a lot of junk there. So then I want to switch. So that's all theoretical stuff. That's just looking to see what we can do. It's a little bit of a, it's not a perfect system because you have to be careful. Those small fragments might actually denature during some of the processing, during some of the library prep. But if it's a genomic DNA sample, they might not find each other, and you might not get a good library prep. So in this case, it's a pure sample. If they do denature, they can hybridize again, and so forth. But we've actually been able to do whole genome libraries as with fragments and inserts that are sequenced as small as 35 base pairs. Okay, to give you an idea of what we can do with a wide range of DNA insert sizes that are very complex. Okay, so cell-free DNA, as I said, liquid biopsies are super exciting these days. Everybody's interested in being able to take a sample of blood, look at the mutations in that person without having to do a true solid tumor or other type of biopsy. And so we wanted to look at cell-free DNA. Cell-free DNA comes, we bought this DNA from BioChain. They have a kit for purifying. They made cell-free DNA for us. And usually the peak is around 165. It's apoptotic DNA. It represents the nucleosomal size of DNA from those cells that's circulating in the blood. And often you'll see a 330 as well. These guys this prep had a bit of genomic DNA that was also contaminating from probably white blood cells in the sample. And then we also did matching genomic DNA, and we sheared it to 377 base pair peak size. And then basically we made 3 libraries from these. We did 250 picograms of cell-free DNA insert, 1 nanogram of input, and then 50 nanograms for our UltraLow kit. PCR cycles are there. We did our standard 2x, 1.5x pre-PCR, post-PCR cleanup steps and size selection. And then these are the cleanup steps that we used for the standard genomic whole genome libraries with the genomic DNA. And what you can see is the libraries are beautiful. These gives me goosebumps when I see them on the Bioanalyzer. But you've got a really nice 295 peak here, and you've got another 330 peak here. These are the mononucleosomal and dinucleosomal sizes that you started with. You end up making beautiful libraries. Don't get a lot of the large genomic DNA present, anything really there in the sequencing. And then both the low input and the higher input gave us beautiful libraries. And then the genomic DNA was a 520 base pair peak of library fragments here as well. So then we went on to sequence these. And we did a MySeq first, just to make sure these libraries were high quality. And what you can see is that the mononucleosomal came out at a peak reads of paired end reads of 165 base pairs and 330 for that small peak, just as we would expect from mono and dinucleosomal sized insert. fragments. Both of them looked very, very similar. And the genomic DNA, I think, was peaking around 300, 310 base pairs or so. And of course, we went on to do high-seq sequencing. And actually, the results from the cell-free DNA look as good or better in many cases than the genomic DNA. So here's the 3 libraries. We actually sequenced 4 libraries on 2 lanes of a high-seq 2500. One was unrelated, so it's not shown. Did 2 by 150 base pair chemistry. We downsampled prior to analysis to compare apples to apples. And then we mapped the reads back to the human genome. And what you can see is our mapped read percentages are really good. They're actually a little bit better for the cell-free DNA. This was unusually low for us. We're not really sure what the deal was here. We looked at it very closely, and it doesn't look like any of the metrics are off. I suspect it probably has something to do with the DNA sample that we got from BioChain, but we're not sure. Unique mapped reads, very good from cell-free compared to genomic. Duplicate reads are a little bit high in this run. And then properly paired were good, and then the insert links were what we started with. So we got about 300 base pair inserts for the genomic DNA, and then 166 on average for the cell-free DNA. So exactly what we would expect. We looked a little bit closer, and at the coverage, you can see the coverage is 13x for the 50 nanogram genomic DNA. It's less for the cell-free. Remember, these fragments are about half the size, so you would expect a lower depth of coverage. But the breadth of coverage, or the zero coverage regions, are very close. And this is, again, whole genome. And then the coverage is very good. So there's very little difference in zero coverage versus the whole genomic DNA input. And the estimated library side by complexity here with the 1 nanogram input is excellent compared to the genomic DNA. So we're making really good high-quality libraries here. We're really excited about this. And then we've got a mystery collaborator to finish up. They're doing micrococcal nuclease chip-seq, okay? And this was their standard chip-seq. They're an origin, they're a DNA replication lab. I'm not going to give you their name because they're trying to give this all out and get this published. But this was their first attempt at looking at this one particular origin, which is marked by 0. And the chip signal is very weak. It's just barely above background. But when they used our kit with our UltraLow protocol and micrococcal nuclease to do the digestion, you can see they got a beautiful, nice, strong chip-seq peak here right on top of their origin. So they're super excited about this, and they're applying it to their work, and they're really going to take that to the next level. Okay, so then just to finish up, so hopefully I've convinced you that we can make really nice high-quality libraries from small DNA inputs. And as I said, you know, I didn't show you the data, but we've been able to generate whole genome sequencing libraries where we can get reads from fragments as small as 35 base pairs. And the key has been our universal adapter, which is this small mini adapter. It allows us to do the proper size selection and separation of the junk, the adapter dimers and adapters, from the actual library fragments. And we've been able to modify the kit and the protocol to work with cell-free DNA and then this micrococcal nuclease digested chip-seq samples. And I would assume, and I hope that it'll be true, that it'll work great on FFPE and other metagenomic samples and so forth, but we just haven't gotten to that data yet. So anyway, I'll stop there. And I guess we're not taking any questions. So
we actually finished a bit early, so we actually have time for Rob for a question or two. Does anybody have a question for Rob about this technique, this new product?
Just want to see data storage stuff.
An RNA kit?
There is not an RNA kit. No, not yet. All right.
Next phase for you guys. Okay, thanks, Rob.
Thanks.